So good afternoon, everyone. Thanks so much for joining us today. We're thrilled to have you here at Millennium Science webinar showcasing Agilent's xCELLigence real-time cell analysis platform. My name is Joe Roberts, and I'm the product manager at Millennium Science, and I'll be your host today. Joining me online, we have also from Millennium Science, Christopher Pickup. So he's our territory manager based in Queensland, and he'll be helping moderate the Q&A and the chat. And we're both delighted to welcome our guest presenter, so Dr. Michael Smout from James Cook University. Michael has a wealth of experience using the xCELLigence for his research, where he began his career studying melanoma and viral infections, but for nearly 2 decades, he's been focusing on parasitic worms and their potential in medical research. He also explores venomous species and is developing a wound healing compound to aid those suffering with chronic wounds worldwide. So this is today's agenda for the webinar. So I'll give a brief introduction to Millennium Science before focusing on the Agilent xCELLigence and walking us through the powerful real-time cell analysis technology. Then Michael will take it away with some really cool xCELLigence experiments, looking at worm wiggles, wound healing, and altering heart rhythms with box jellyfish venom. And then finally, we'll end with a Q&A, and then all attendees here today will be entered into a draw for an Amazon gift card, and that will be drawn after the webinar, and the winner will be announced by email. So Millennium Science. So Millennium Science was founded 25 years ago, and our mission has been clear. So it's to ignite biomedical discoveries through the power of revolutionary technologies. We're proud to support the research community by distributing cutting-edge instrumentation and reagents to scientists across Australia and New Zealand, enabling groundbreaking work in everything from cancer biology to infectious diseases and beyond. So at Millennium Science, we're proud to be recognized as a leader in cell analysis. We represent some of the world's most innovative life science brands, such as 10x Genomics, Mabtech, and of course, the focus of today's webinar series, which is Agilent. These technologies empower researchers with a comprehensive cell analysis workflow from cytokine profiling to single-cell sequencing and real-time label-free analysis. So today's we'll be zooming in on the Agilent xCELLigence system. So this is a powerful tool that's changing the way we study live cells in real time. So let's dive into the heart of today's topic, which is Agilent xCELLigence and its real-time cell analysis. So this is a cutting-edge technology that allows researchers to monitor living cells continuously, label-free, giving real-time insight into the cell's health, behavior, and responses to treatment, all without ever lifting the lid on your plate. It's fast, flexible, and incredibly informative, and changes the way how we study cells. So there are several different xCELLigence instruments which have evolved over time to suit the needs and application of every type of lab. Between 2008 and 2012, ACEA and Roche launched 5 xCELLigence instruments with different types of throughput for different types of applications, many of which are still being actively used today. Following the end of the partnership with Roche, ACEA developed a small xCELLigence instrument that incorporated an iPad to operate and a second-generation cardio system. In 2018, ACEA became part of the Agilent portfolio, and since then, a further 3 systems have been launched: the ePacer, the eSite, and the RTCA BioSpar. So this highlights that there's an extensive range of products for the xCELLigence technology that suits every type of lab. So the xCELLigence offers an automated walkaway system, allowing you to focus on the science instead of the manual, time-consuming endpoint assays. So the workflow involves seeding cells into the bottom of an E-plate before placing them into an xCELLigence instrument, which is situated inside a tissue culture incubator. This gives you time to relax, read publications, or prepare follow-up experiments, all whilst the xCELLigence takes readings of the cell's health and viability in real time under physiological conditions without any hands-on involvement. The beauty of this technology is that the data analysis is also automated, and since the cells are not labeled, you can perform further downstream experiments on the same cells. So let me take a moment to talk about the impedance, which is the principle of the operation behind the xCELLigence. So this is a diagram of a side view of a single well, which is covered in gold microelectrodes at the bottom of the well. These can detect the flow of electrons, and as cells are added, this impedes the electron flow and allows us to detect changes in cell number, morphological changes, adhesion strength, and barrier function. And these changes in impedance are calculated into a unit that's known as the cell index. So here's an example of a typical data response. So at the beginning of the experiment, when the wells are empty, the cell index is 0. When cells are added to the well, the initial adhesion and subsequent cellular proliferation are recorded in real time by the xCELLigence. The biosensor technology measures the cell attachments by using the gold microelectrodes embedded at the bottom of each E-plate, and the growing cells obstruct or impede the electron flow. So following the addition of a toxic agent such as a drug or a CAR T cell, this can kill the cells and the membranes are compromised, they shrink, detach, and then these results will result in a cell index change. So the xCELLigence technology has a solid publication track record expanding over many years. Only last month, the xCELLigence real-time cell analysis instrumentation was used in an FDA approval of a CAR T-cell therapy. And finally, the beauty of the xCELLigence system is its versatility. So it can be seamlessly incorporated into most areas of cell biology research, whether you're focusing on cancer biology, immunology, infectious diseases, or drug screening. So the xCELLigence provides real-time, label-free insight that helps take your experiments further. And with that, I'm delighted to hand over to Michael Smout, who's going to be sharing his insight in how he's been using the xCELLigence to maximize his research and uncover some truly fascinating biological responses. So I'll hand over to you, Michael, to take away.
Wonderful. Thanks, Joe. All righty. All right, so just a quick brief introduction to me, Michael Smout. I'm from Australia, from the north of Queensland, from Cairns. Our lab's nestled in the side of the rainforest there, with of course the Great Barrier Reef and all that comes with it, and of course the various other things in Australia that can kill you. So that certainly allows us a great range of various different things to test on xCELLigence and other assays that we do. So I'm going to be running through a couple of uses I've put the xCELLigence through with saving diabetic feet with worm spit, our granulin peptide, our X-worm worm dancing assay, and then cardiotoxic components of the sea wasp box jellyfish. So a quick update on the wound healing basics. So everybody wounds themselves, and usually a bandage in time will heal that wound. But sometimes this wound healing fails. And this leads to chronic wounds. These are wounds that just don't heal on their own and can take months and months of hospital treatment to treat. 1 to 2% of the world will suffer from these wounds, and they tend to be more prevalent in smokers, diabetics, the elderly, and military personnel. So what's the solution? Well, some kind of Wolverine supercharged wound healing ability would be nice, but since we don't have that, we're going to have to work on something ourselves. So we might have a really unusual solution, parasitic helminth worm secretions. So I work on Opisthochus viverani from Thailand. This is a worm you get infected with by eating undercooked fish. And it'll crawl up into your liver and be a centimeter long leaf-shaped flatworm that'll live there for decades. It'll be munching around your liver wall, and all the while it's healing up these little wounds as it goes. And the way it's doing it is with this granulin peptide that we discovered. This is a really unique protein that is in everything from bacteria to plants to us, and it's evolved in development and wound healing. And it's got this fascinating structure where it's twisted upon itself, held together with disulfide bonds, which are those yellow jagged lines, holding a 10 kilodalton protein together, but it performs like it's 1.5. So what we're able to do is show that this protein had wound healing capabilities, and then further optimize it by making a 24 amino acid peptide that we're able to synthetically make in the lab, named GP4A. And so what we're hoping to do is to turn that into a wound healing cream, something maybe named Helminth, to be able to improve chronic wounds and all wounds ultimately. So we're exploring this GP4A worm peptide as a wound healing cream with a range of different assays. I'm going to be running through proliferation, scratch wounding, mouse healing, and human skin healing. So as Joe went through, this is the xCELLigence system that I use in a lot of these different assays that uses impedance to measure the cell number in real time. And here we're doing a simple proliferation assay, add the cells, watch over time, and you see an increase in our 1.5 and 6 nanomole of testing our full protein here to increase over the controls. And so while proliferation is the first step in screening for wound healing agents, ultimately the scratch assay is one of the more routine assays we do with cell lines. And conventional scratch assays, you grow the cells to a monolayer, you scratch with a pipette tip, treat, and then watch for that wound to heal. Now, as you can imagine, scratching cells manually with a pipette tip is not particularly reliable or easy to do at large scale. So that's where the AccuWound device comes in, which is a system with 96 silicon fingers that works with xCELLigence plates and is able to do 96 relatively consistent scratches all at once. And we'll just go back there. This shows roughly the cell monolayer there. And then once that you've applied those silicon fingers, it creates a scratch wound. And then that over a period of time, those cells will proliferate and migrate across that wound. And that's what we're going to be monitoring with xCELLigence. Here's what the raw cell index looks like when we seed with 12,000 keratinocyte cells. So the circles there are the gold electrodes. And then here the cells are stained purple that to allow you to see the cells easily. So that's growing the cells to a monolayer. We scratch and treat. And so you can see those gold electrodes are mostly revealed. And then over a period of time, those cells multiply and migrate into that wound, causing an increase in that cell index. And there you can see with various different treatments, we have our control there in blue and red and green with our treatments, improving that wound healing process. You can choose various different time points, and I've chosen to focus on the 4 to 36 hour time point because that gives the biggest window of activity time to allow the treatment to show its colors. So here I'm going to run through our GP4A, our active granulin peptide movement, and then GSP control. So that's a sequence with the cysteines taken out, and then the same sequence as the rest of GP4A and just scrambled into a random sequence. So to give us the same, almost the same sequence with a random structure there with GSP. So here you can see our GSP causing very little change to our controls, and then our peptide mutant causing an increase in the wound healing over time, getting above that 20%. And the important thing to note here is not only we're getting rise, we're getting this bell curve where after you get a peak of growth, you get a steady decrease, but it is over many fold, a large range of concentration. When we look at the 95% confidence range there, we see we have our 22% peak at 148 picomoles and a wide EC50 range of 10 picomoles to 419 nanomoles there. So there's a wide range of potential therapeutic doses. And also what you can do in these cells is increase the sugar concentration in the media. And that is a very crude way of replicating diabetes on these cells because they slow proliferation and slow migration as they grow. And here we can see a similar response that we're getting over a similar range and almost as high, up to 19% at 25 picomoles. And this is at 24 millimoles of glucose, which is considered the beginning of the diabetic range with these keratinocyte cells. Looking at these two concentrations, you can see how similar they are, having quite a lot of overlap. So suggesting that our treatments would be effective in people under normal conditions, but also in potentially diabetics, who are one of the high groups who suffer from these chronic wounds. So here's our mouse wound healing assay. Here's our little mouse here. They're anesthetized and given painkillers. And we create a 5 millimeter wound between the ears. And that's a full depth, skin deep wound. And that's important that it's between the ears because the skull serves to splint the wound open. Because normally a mouse, a lot of the healing will happen strictly by contraction. So that's important that the wound's splinted open so we can monitor it healing. There in a methicellulose gel, a treatment's added, and then a spray bandage is used to seal that wound. And then over a period of days, we photograph the wound and monitor that healing. While we've got various time points, we tend to focus on day 4 because that's when a lot of the cells have had a chance to replicate and migrate into the wound. And here we see our various different controls there, all sitting around similar levels. Our full protein, that full granulin protein in orange, with 27% above controls. Regranax, 22%, and it's the only biological treatment on the market for chronic wounds or for wound healing in general. And then we've got a native peptide sequence, so that's the same sequence from the worm, but 24 amino acids with 19% above controls. And then we set, here's our GP4A. And why it's so unique is that we took out one of the prolines, and these are amino acids that can cause a kink in the molecule to twist it one way or the other. And sometimes they can twist it the wrong way. And when we took out this kinking proline, we're able to not only improve our healing 43% above controls, it was able to be significantly improved above Regranax, 21%, almost double the healing rate of Regranax, which we were particularly excited by. And then we go to one of the most incredible assays that we've got available before we actually test in live humans, is using human skin. So from weight loss surgery, you're able to grow that skin in the lab and be able to do wound healing assays on it. So the centimeter square pieces are grown in the lab. Extra cells are added to those skin pieces. They are human primary keratinocytes. They develop and differentiate when exposed to air, just like our skin layers. And then we're able to, with a biopsy punch, create a 4 millimeter diameter wound, peel off that epithelial layer, and create a wound-like situation. Then, so here's what a day 0 wound looks like there. And you can see that the stained cells in purple, and then the white patch in the middle where we've created that wound. And so then we're able to look over a period of time and see what our treatments is doing. So here's that schematic again. You can see the piece of skin tissue exposed to air, but it's surrounded with a lake of media that provides the cells with nutrition. And here's an example of a controlled treatment and then an effective treatment in purple. And you can see how that wound area is shrunk. So when we look at the real images, here's our day 0, and then here's various treatments, our SFM serum-free media control, as well as 2 and 20 picomoles of GP4A. And you can see how much the smaller that wound is, especially in the 2 picomoles where it's completely healed or virtually completely healed over. When we measure these various time points, we show a significant improvement with both our 2 GP4A treatments. And so that was particularly promising. And I won't go into all the details, but we've done follow-up experiments where we test skin pieces from different patients and we get a similar response. So what are our future plans? Well, hopefully when I'm old and grey in 2060 and I get a chronic wound, I won't need to get an amputation. I'll be able to get a little bit of Wolverine in a cream, maybe named Helmential, and hopefully be able to heal that wound up and keep my limb. So that's certainly what we're hoping for in the future with GP4A. Now moving on to one of the exciting uses we've developed for xCELLigence. So xCELLigence usually is used for cells, but we're able to use it in all sorts of wonderful ways in our parasite lab, and this is our X-worm worm dancing assay. So a little background on parasitic worms. So they're not as bad as Hollywood movies, but they can be very devastating, especially in low nutrition areas with high worm loads, causing a range of side effects in everything from our companion animals, cats and dogs, to sheep and cows as livestock, as well as humans, depending on what region of the planet you're on. These parasites cause morbidity to billions of people and livestock, and they can be quite severe infections, costing billions. A particular barber pole worm here costing 3.7 billion to the sheep industry. And resistance is actually becoming increasingly common in a lot of these livestock treatments. And human parasite resistance is looming, as we've seen some worrying signs happening at this stage. And there's limited high throughput monitoring methods. Only a few have been developed over the last decade. So we've got our potential solution, our xCELLigence worm real-time motility assay. So as I mentioned, it uses the xCELLigence system, just the same as what you do with normal cells, but rather than having cells bind to the base of the plate, we put worms in there. So this is an adult hookworm, roughly 1 centimeter long, and it writhes around in that plate. So I like to think of these worms writhing around like worms in a bucket, that sometimes they touch the bottom of the well, sometimes they writhe up and swim in the media. And so what they're doing is constantly changing that amount of contact. And so rather than looking at the simple cell index like we did when we were looking at cells, what we're looking here is the variation in the output. So here I've marked in the orange the full motility. And so that's that high variation of the worm wiggling about. And then when we treat these worms with different treatments that cause reduced motility in red, this is a paralytic drug, you can see how much that variation drops. And then with dead worms in green, the immobile worms, it's reduced again. And that variation is very similar to the empty well down in yellow at the bottom. Now, just to reinforce that the raw cell index here doesn't matter. I've shifted these so you can see them all. Normally, when you run the experiment, they'll all overlap, and then it's this variation we're looking at. And what we do is use the standard deviation of that range over time, and we're able to turn that into the motility index, something more conventional similar to the cell index that you'd be using for cells. So here we have no drug in black, and you can see that while it bounces around a little bit, it broadly stays stable over a period of 4 to 5 days. And then with increasing concentration of praziquantel, which is the drug for treating schistosomes, which we have in this particular experiment, you can see that there's an increasing reduction in the motility index. But also intriguing, what we saw was there was if you didn't kill the worms, a lot of the time at these sublethal levels, like that green curve, or especially the cream colored curve there, you got a recovery. And even in sometimes with this cream curve, you get a hypermotility. So that's suggesting they're moving much more than what the controls are. So hyperactive, if you will. So this is also something that's been confirmed by different systems that use visual identification. They also see this low level causing a hypermotility. And as much as I'd love to lay claim to it, I can't claim the wonderful name discoschisto that these hypermotile worms are given. But it's particularly amusing to think of them dancing there. So once you've created this motility index, be able to do conventional dose response type assays, creating EC50s over time, IC50s over time, and then you're able to show that, for instance, in this instance, the IC50 increases up to about 48 hours where it plateaus from then on. And so this is particularly useful for creating drugs because you have a certain amount of bioavailability and knowing how regularly to dose a drug. And then this assay can also be used for detecting resistance in worms as well. And detecting drug resistance is a particularly novel application that this can do well. It's good for a whole range of different life cycle stages and species, both roundworms and schistosomes, which are platyhelminths. There's egg hatch and L3, which is the microscopic infective stage, and then the adults, which are range depending on the worm species, but somewhere between 1 and 2 centimeters long often. And so here just shows a couple of different range. Pretty much, if it actively moves in media in a well, and you can fit it into a 96 well, the xCELLigence will be able to measure it. The only life cycle stage we weren't able to measure was this somnule stage, which is a very slow moving blob. But luckily there's conventional fluorescence systems that are able to monitor life and death. So adding xCELLigence to the conventional systems are able to vastly improve how we can monitor worms. And then just recently, one of our students has been able to fully optimize for her species of worm here. And so here we've got a whole range on the x-axis. We have the parasite density because these are microscopic L3 infective stage worms. And you can put a whole range from up to hundreds to thousands of worms. And then also the media concentration, testing both PBS and DMEM. So ideally, these worms would probably be in water, but because the xCELLigence system does need a low level of salt to work, we tested all the way down to 3% media, and we're able to get a good response there. So you can see, while there's a whole wide window that was effective in these two different medias, you can see those red circled areas with the dark green have the highest response there. And you don't need to understand the full details of the SSMD metric, but largely it's a proportion of the variation and the distance between controls and the dead worms. So it's a particularly great way to measure a whole wide range of worms, and you don't need to know much about the worm activity before you test them. But now we're also going on to the cardiotoxic of the sea wasp box jellyfish. Firstly, simple toxicity, and then heartbeat rhythm disruption. So Chironex leckeri, the Australian sea wasp box jellyfish, found throughout Northern Australia, the most venomous animal on the planet, with about 120 meters of tentacles, with only needing 2 meters to kill a human. It's got various toxic activities. It will damage skeletal muscle where it envenoms you, but most of the lethality comes from this cardiotoxicity, the ability to rip apart heart muscle cells. So we've done a whole range of different assays looking at the simple cell toxicity. So that's similar to the cell growth assays we did, but here we're looking at toxicity, where you grow the cells, you add the treatment, and then in proportional to the concentration of venom, you get an increasing toxicity. And each of the dots here you can see is 15 seconds. So you can see that that purple curve there is completely killing within a couple of minutes. So that's just an indication of how quickly these things can kill. So we focused on one of the toxins. There's various different toxins within the venom, alpha, beta, and gamma. And alpha and beta are the particularly potent ones, and they're many-fold more potent against heart muscle cells than skeletal muscle cells, like I mentioned before. So here's our simple cell toxicity graph with the various concentrations, showing that the more you add, not only the more cells it kills, but the quicker it kills as well. And so for the next part, we're talking about heartbeat rhythm. I'm just going to be focusing on the areas circled, squared in that yellow square there, going from 200 picomoles and below. So each of these data points, because we're using the eCardio system this time, which is able to grow cardiac stem cells in the dish and actually get heart cells to beat like a normal heart would. So it's incredibly valuable amount of data. And so these 15,000 reads are doing a more advanced version of what my worm dancing was doing. It was able to detect the contraction of these heart cells. And so here's the raw output. And when that's processed, it's able to give an output somewhat similar to what a conventional heart monitor would give with an EKG. So when we're looking at just a mere 2 minutes after treatment, we're able to see that our 200 picomoles completely ablates the heartbeat, and the 20 picomoles has a very minimal beat that's quite irregular. And then, as you can see, getting up to a no treatment control, the effect is gradually lessened. But even at those low levels, 2 picograms especially, you can see how close the heartbeats are together. So suggesting that the heartbeats are beating faster and quicker. But after 90 minutes of toxin addition, we're getting complete recovery from these low doses that showed quite a marked effect. And this is actually what we see in the real world. When somebody's envenomed on the beach, if they get a sustained amount of CPR, which can often be up to 45 minutes, they have been known to recover. So it is particularly interesting seeing the real world effects of this venom matching what we see here with the cells. But even those low levels, even the 200 picomoles, where we saw no measurable beat after 2 minutes, we're seeing almost full recovery, despite the raw cell index being 35% below controls there. So it's particularly interesting that these cells seem to be shut down. And that's part of our other work that we've showed metabolism is inhibited and with these treatments, and then the recovery happens. And so that's particularly useful for potential therapies. So we're able to go through, create conventional IC50s, which is that raw, the normal toxicity, and then also IC50s of the amplitude, showing how quickly they're beating. And so we're able to show that the IC50 amplitude was several-fold for all of our treatments, but especially for alpha. That's the A mark there. It was 13-fold more potent in knocking down this amplitude, in affecting the amplitude than the raw toxicity. So suggesting there's a whole complex of compounds here. But also we have two new, I mentioned we've previously found alpha, beta, and gamma just with simple toxicity, but we also found X and Y peaks, which were very minimal and we hadn't noticed before in our simple cell toxicity tests. And these were reasonably potent with knocking out the cell amplitude. So it's suggesting that not only is the toxin destroying cells, but it's able to affect the amplitude in completely novel ways. And so this all adds to the complexity of creating treatments, but it's certainly fascinating to study. So to summarize, we've got a granulin wound healing peptide that's a promising and potent wound healing agent that we're hoping to develop into a wound healing cream. We've got our worm dancing X-worm assay, which is highly variable for worm species, life cycle stages, and different medias. And the sea wasp box jellyfish toxicity that's highly toxic on heart muscle cells and even more potent when heartbeat analysis is incorporated. Thanks to all the various teams who've helped us over the years to get all this wonderful range of data. Any questions?
Thanks, Michael. That was a fascinating insight into how the xCELLigence system can be used for many different applications. I love the concept of a schisto disco, but I'm not sure I'd like to attend the disco.
I wouldn't mind being a bit like Wolverine.
Yeah.
That's certainly the dream, isn't it?
Yeah. So I'll open
Well,
the floor to
hopefully,
any questions. So if you've got a question, you can put it in the chat or raise your hand if you want to speak directly.
Yeah, we've got one from Donna McIntyre already.
Cool, great.
Michael, can you see that at all?
yeah, absolutely. So Donna says this is very interesting research. As somebody who works in clinical trials, what is the timeline for progressing the wound healing research from phase 1 in human clinical studies? Great question. We're currently looking for funding partners to help us get there. We've got a plan for 2 to 3 years to get into phase 1 trials. And then from there, it would depend on how it goes. Some of the diabetic trials can actually overlap with phase 1 and 2A, so there can be some time savings there, but it would depend on how the results went. So yeah, maybe 5 years after we started the phase 1 trial if things went well, but of course these things are heavily limited by funding.
Cool.
Thanks so much. Any other questions?
I had a question, Michael. So yeah, you've used the xCELLigence, you've pioneered like the xCELLigence, how you can use it for different applications with the worm wiggles. Have you got anything else in the pipeline that you're thinking, how that you can use the platform?
Well, it's not so much stuff we've pioneered. We're looking at combining some different cell types with these transwell, effectively, inserts that go into the xCELLigence wells, looking at fibroblasts and keratinocytes together, because they're what both in the liver can cause, likely be the precursors to cancer, but then also in the skin wound healing, combine together to heal that wound. And so getting a more thorough response, trying to recreate the full system by having either cells going together directly or cells separated, but sharing the same media with the transwell. Hoping to expand what we can do there, and then doing that with the scratch wound healing to try and get a better appreciation for how this is all behaving.
Cool.
We've got another question from Patrick Tang.
Great. So Patrick asks, how does the worm wound healing properties fare compared to commercial products? So that's something we're exploring, but in our mouse assay, the major commercial product at the moment is Regranax, at least the only biologic on the market today, and we were almost double its potency at 4 days, so we're particularly happy with that. But the major market for these treatments is chronic wounds, and they're very difficult to study in animal models. So until we got into more advanced clinical trials, we wouldn't be able to compare them directly. But so far, we show promising results that may be more potent than Regranax.
Any more questions, or we'll wrap up there?
Yeah, I just
Any
wanted
more?
to say a big thank you to Michael and Joe for presenting today. Mick, yeah, your research is absolutely amazing. It'd be amazing to see it on the market one day.
Hopefully, we can get it to the shelf.
Yeah, and I just wanted to remind everyone that you're all in the draw to win a $50 Amazon gift voucher that'll be announced via email after this. And we'll be setting up the following up seminar soon. So we'll look out for some invitations to that one. Thanks very much for attending today.
Cool. Thanks, everyone.
Thanks for coming.